conditioner?
How do you feel like? It's okay? Temperature is fine? Okay. You want to come closer?
Yeah.
I think the closer you are, the better. Right. We lost some participants.
Classes are there in the morning.
What do you say? Oh, there are classes.
Classes going on in the morning.
Okay, got it. Yeah. Okay, sounds good.
That's why we're actually planning for initially from 4 to 6.
That was the reason.
We
Okay.
don't have classes.
Right, got it. So that's okay. Well, I think the idea is that in the morning there is the the lecture, and there is a time to breathe, and then in the afternoon you make the experiments. I think for 4 hours was a bit difficult because I think for listening to, I mean for everyone, I guess for the speaker and also for the participant. So I think I would like to start. Maybe it will get some time for the people to join also. So what did anyone measure the MSD of yesterday? First of all, what did you see yesterday afternoon? Then I'll go into the meat of the lecture of today. What did you measure this afternoon? Yesterday afternoon, what did you see?
Sir, actually we tried to measure it. I mean,
yes.
So the difficulty was that first of all we couldn't find the track in the extension.
Yes.
When we found it, but the video wasn't. We found a little difficulty uploading the video, as I mentioned.
Yes.
So
you needed to convert it. But the idea is that the typical movies that you have with your cell phone, you cannot upload them on Fiji, so there is a need to translate them from one format to the other. That's right. So this will go through that. You can do this online. You need to,
that also.
And it was not working.
Yeah, not okay. AVI. It was in MP4, recorded in MP4.
Yes. Yes.
Tried to convert it to AVI format.
Yes.
But still there was.
Yes. So you should try, and this will go through this. TIFF should do. Because TIFF will be open, and then you can use the TrackMate. I mean, all these trials and errors, if you manage to do it at home between the session, I think that's the best, and that's the way also. Actually, usually that's what most of the people are doing, is that they go through the test of error. But exactly the point. I mean, first of all, you need to upload Fiji, then you need to have a conversion with the right format. I would advise TIFF, and after you, if I may say, women have the priority. Yes. Little motion. How would you qualify the motion?
Moving very slowly and very small distances.
Very small distances, very slowly. I agree with you. Did you do anything special to have this motion? No. Right? You want to say something? How would you qualify with word this motion? You didn't quantify, but just with eyes, with bare eyes.
With eyes.
Which words would you use to describe it? Like this, Mike, like this, right? Is there a word that comes to you when you observe that or not?
Diffusion.
What do you say? Diffusion. It's an already very educated word, right? But just, I would like that we rotate in the room. I'm sorry, I like your answers, really, but just I would like that everyone participates. Yes. It looks like vibration, yes, yeah. It looks like vibration. What else? Which other words comes to you when? Randomness. There is an idea of randomness that you need to demonstrate, and you need to show that it is correct or incorrect, but there is an idea of randomness. Okay, so far so good. You remember also that yesterday I highlighted the fact that, in fact, this motion was essentially at low Reynolds number. This was the end of the lecture yesterday. What does it mean, low Reynolds number?
High viscosity.
High viscosity, right? So it means that even though it's moving all the time, and this is very counterintuitive, it's moving all the time, nevertheless, there is a high viscosity, meaning that, inertia is totally negligible in this region. Okay? And the idea was that you get a sense of the fact that because of a scale, not because of chemistry, one of the participants was asking me, I think it was you, maybe another person in the room was saying this, oh, it was you probably. One of you two, where you were telling me that somehow it's very different from a chemistry approach, which was surprising in a sense, because then I just considered the size. And indeed, this is the size which is the argument, actually. That's essentially all driven by the size. And I went through different scaling arguments, one after the other. One was order of magnitude of forces, which was the ratio of the energy over the size. It gave piconewton. And you have one expert of optical tweezers, so please, ask any questions about how to tune this type of, how to trap these beads and how to manipulate them and play also with these fluctuations. Then I went through the diffusion constant with the Einstein relation, where I was showing that it was the ratio of the energy over the friction. And then I was going through the number, which is the Reynolds number, which evaluates the ratio between the what and what? Inertial force over the viscous force. And we showed that it's 10 to the minus 4. So, I was just reporting it's 10 to the minus 4, meaning that everything is dominated by essentially, viscosity. Right? And I was insisting that it is counterintuitive, because it's jiggling around, as you saw yesterday, right, with the full scope, and yeah. We haven't shown the randomness, so we're going to show the randomness probably either this afternoon or tomorrow, but there is something that is random around it. Okay. So far, so good? Any comments, questions before we move forward? Yes?
Sir, even if it was vibrating at its own place,
yes.
It's called Brownian motion, still you said that we have to make it stable so that there is no effect of the moving fluid.
Yes.
But still, it was going from one direction to other.
Yes, right.
Was it that? Can it be ascribed to Brownian motion or maybe something?
So the flow that many of you were complaining about, and this afternoon we're going to face it again, whatever we do, and actually now you are used with full scope. I think most of you now you know how to manipulate it. You have the three objectives. You know how to interact, to make the interaction with your cell phone. If you could this afternoon make as much as possible the due care about stabilization so that it's perfectly flat, I think that would prevent this global motion. This global motion, many of you had the intuition of where it was coming from. All the particles were jiggling around. The 3 micrometers that I gave, the 6 micrometers that Pramod gave, they were all jiggling. But there was a flow. What was the origin of the flow? Where was it coming from, the flow? It was a bit, yeah. So then this afternoon, if you can repeat, actually, you will try to make it as flat as possible. And what I would like to do is to try to give, and together with Pramod, some beads that you would put in your sample so that you can calibrate. But somehow you need a ruler. The ruler, you have them in the experiment, but that's it. Last question, and now we go next to the lecture of today. What was the qualitative difference between the 3 micrometer beads and the 6 micrometer beads? Without academic knowledge, just by bare observation, neutral observation. What was the difference between 3 micron and 6 micron?
So we are getting like a gravity effect in this size of particle.
Yes.
Gravity effect, something like gravity effect if we increase the size of beads,
yes.
We are getting gravity effect of environment like this.
You could see the gravity, right? So you could see the 6 micrometer beads. What was they doing?
Gravity is effective there.
Yes.
Decrease the size of particle, there is a negligible of environment effect.
Okay, so basically in your movie, instead of having jiggling around, what did you see?
So degree of motion that is more in case of smaller particles and it's slower in the larger.
It's smaller, but when you say, I heard gravity, right?
Yes,
So gravity is, okay, they're smaller, but on top of that, did you see them sediment? So gravity means what? Gravity means that they were going down.
Yes.
That's what you saw?
Yes.
Okay, right. So before that, you were seeing them jiggling around or not?
Yes.
Okay, right. But to a lesser extent, that's what okay, right. So what we'll do either this afternoon or tomorrow, it depends a bit how the dynamics of today goes, but basically what we'll try to do is to quantify this motion of the 3 micrometer beads, the 6 micrometer bead, and compare it to the Einstein relation. You remember the Einstein relation was the ratio. Come closer. There is a lot of seats here. Come. Anywhere. 3 micrometer, 6 micrometer. Basically, when you compare the size of the particle, we should be able to compare basically how the diffusion constant is going to be affected. Right? So this would be an easy way for us to compare and to get a sense. Right, so we're ready to go. If there is no more questions, yes.
What is the scientific significance of that?
Of what?
I don't know the error bar.
Yes, okay. So you mean not the biological insight, you mean more the if you want to be rigorous in terms of error, right? So we'll go to that to some extent this afternoon, and most probably tomorrow afternoon, is that whenever you get a motion of a particle, right, how can you estimate the error that you make in the measurement? And there are some clear rules of how to estimate the error you make in the time, the error you make in the space, and then doing this, the error you make in the overall measurements. So that's the error bar that you will get next. It is different from the biological insight, which I was highlighting yesterday, that I want to repeat one last time, and then we'll move forward. Why I wanted to highlight diffusion is that whatever you do, and returning to chemistry, it's key to consider the chemistry, obviously. But in addition to this, you have to keep in mind that any object of the nanometer scale or the micrometer scale is submitted to Brownian motion, basically. It cannot escape it. It's a given. It's somehow, it's a rule of matter that cannot be counteracted. And so in that context, bear in mind that if you have an expression of a gene that depends on a very low number of events, which are random, you will have the transcription factor, which is the protein, the complex of protein I was mentioning yesterday, which will be wandering around and then eventually found the target gene and then bind. But it will go through Brownian motion. Look, 1 micrometer, 3 micrometer yesterday, it was making a little jiggle. Go 3 orders of magnitude lower to 3 nanometer. 3 nanometer, it's going to be like jiggling around like crazy, right? You wouldn't be able to see it with your bare eyes. So imagine, again, a transcription factor going to a gene. It's going to be an extremely noisy system. And throughout the biological system, this noise is present throughout. So it gives, if you wish, some richness in the system, but also some noise. And it's an intrinsic property of living matter. And that's why I wanted to start by this yesterday, is that it's a given to have this Brownian motion as a given. Come closer, come closer. Closer, 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 closer. I mean, you see it, yeah, sure. Right, so now we are ready to go. And the lecture of today is going to be a preparation of this afternoon. And now we are really going to go to active matter. So now we are going to kind of real shift to biological system. But bearing in mind that the passive matter is an intrinsic property of all this, and we cannot escape it. So when I say active matter, I just want to say that it's different from inert matter or passive matter. Active is in the opposite to passive. Living matter, I leave it at the moment as a vague definition, right? Living matter, you can imagine that this is living things. You see something, it's living. Anyone wants to define living matter? How do you recognize that something is living? It can move actively, reproduce. It can replicate. But you have two definitions which are very different. A soap bubble can divide into two. It's an active matter, living matter, or it's a passive matter. A soap. A passive matter, right? So basically, right. And moving also is a definition. It's moving, living matter, it's something moving. Right. So I will make living matter, active matter, these two elements as categories. I will leave them open, and the more we move in the week, the more probably we're going to get some sense of what they have. But here, you agree with me that this is in distinction to inert matter that we saw yesterday. And so we need to invent something to make it active. So for those of you who were trained in science, whatever field of science you were trained on, how do you go from inert to active? What do you need to go from inert to active? You need energy, right? So basically the main difference between both of them is going to be energy provided. So you need to give energy to the system. And the point is that this is one of the key properties of active matter or living matter, is that there is energy source. What is the energy source of living matter? ATP, right? So for those of you who are not familiar with, I just want to remind you what it is. So the energy is going to be given by the hydrolysis of, to say it, the first element, there are going to be some other sources, is adenosine triphosphate. Essentially, you transform the ATP into ADP plus P. And this is going to give you a chemical energy. To give you a metaphor, well, in a car, and this is a wrong example because a car is at high Reynolds number, so basically you have inertia, so basically it is just a metaphor for the sake of the metaphor. Basically, you are going to use energy, chemical energy, and transform it into motion. So it's a chemical energy that is transformed into mechanical energy. So it's a transformation of energy from, this is chemical, and eventually you're going to transform it into a mechanical energy. And if there is something that we care about, is essentially the conservation of energy, right? So it means that if there is a given chemical energy that is given, I should have the mechanical energy with a given unit that is going to be conserved. Conservation of energy has to be, given. I invite you to read a very famous chapter by Richard Feynman, the famous physicist, who explains what is conservation of energy. It looks like it's simple. It is not simple. And he explicitly says that somehow the concept of energy is a very tricky one. And he takes the example of, I think he takes the example of a bank account or something, that you know how much you put, you know how much you take out, and somehow you cannot take more than what you have in your bank account. At least that's a recommendation, basically. It should be equilibrated. So energy is like this. If there is chemical energy that is giving you a given amount, the mechanical energy...
Is it different from the first law of thermodynamics? That's what he's saying?
So his only point is the way of saying of defining energy with words. It's Feynman style. So basically he wants to say, force, I know what it is. If I'm applying a force on this chair, the chair will be moving. The point he has in these lectures in physics is the way of saying energy is a fuzzy category. I mean, it's extremely difficult to grasp. So he proceeds to say it's a kind of conventional number or units, this unit. We know how much we put, we much how much we should get. And we know it's conserved. But he describes it very honestly. So it's more, if you wish, conceptual, his approach, rather than very rigorous in terms of communication. But I think extremely inspiring. And it's in his lectures in physics, actually. So if you give a given chemical energy, you're going to get a mechanical energy that is going to be given. I'm insisting on the point that this is the source of energy. You're going to see that I'm going to talk about today about another source of energy, which are going to be the NTP, and NTP is going to stand for ATCG, and you will understand in a minute why I'm going through that. So it's again triphosphate, and here it's adenosine triphosphate, and you recognize ATCG. ATCG stands for? That's right. That's right. So basically you recognize here each of the bases which are going to compose the genome, and it's not an accident that somehow you have also an energy source that is going to be here. I will go back to that in a moment. But the point that I want to highlight is that you provide an energy, or the cells have an energy which is indispensable actually for their life, which is essentially a chemical energy that eventually is going to be transformed into a mechanical energy with the concept of energy, which is a very abstractive one, but very operational one. I would like you to guess what is the order of magnitude of the energy of ATP hydrolysis. Back on the envelope. Just try to make the same reasoning as yesterday, which was the scaling laws. What was the energy of a particle? KT, right. It was fast, it's good. So basically it's KT. what would be meaningful for ATP hydrolysis or an energy source to bring something new? Try to think about it. Just scaling arguments. So basically not sophisticated calculation, but if you want the system to have something more than what inert matter could do, what would be the order of magnitude of the energy hydrolysis of one molecule of ATP? So you say 10 kT. Why do you say 10 kT?
Order of magnitude.
Order of magnitude. So it must be very abstractive for those of you who see that for the first time. Back on the envelope, when you make scaling argument, you say, okay, if I have an energy which is kT without me doing anything, if I want to do something more, back on the envelope, it will be a factor of 10. It could be 100, but you are in scaling argument. So somehow it's like, you make a guesstimate that is not unreasonable. So it's about 10 kT. The factor of 10 is meaningful. It means that if I have a particle that is moving like this, if I provide an energy that is going to be 10 times larger, eventually I'll be able to compete. Because it's more. I mean, it's the metaphor of Feynman, right? If I'm 10 more, provide 10 times more energy, I will be able to outcompete the kT. So this is the first source that I want to say is that there is an energy that is provided. The fact that there is an energy provided inside the system, that somehow the cell is providing this energy, is going to drive the system in a new category of physics that you may be familiar with, which is called out of equilibrium physics. So this, I don't know how much you were exposed for those of you who are with different backgrounds, but because we are in this, the physics is going to be a physics out of equilibrium. This is fundamental because it means that somehow you cannot use the traditional equilibrium physics that you've been learning. I've been learning also when I was a student. It's a different physics. In principle, rigorously speaking, you cannot write in this type of system energy. You can have estimate of energy, but writing a careful energy and minimizing an energy in out of equilibrium physics could be forbidden. So in principle, it opens a new field of physics, which is extremely rich in terms of statistical mechanics, which is out of equilibrium physics. So far, so good? Yes?
How is it different from non-equilibrium?
Is it different from?
Non-equilibrium.
It's the same. It's the same. It's the same concept. Non-equilibrium, out of equilibrium or non-equilibrium is two. Yes. I mean, basically, non-equilibrium is the negation of equilibrium. Out of equilibrium is how far you are from equilibrium. There are some attempts to define how far you are from equilibrium because you can be a little bit far, or even more further away. But the idea is really the same: is that you are not equilibrium. So again, minimization of energy could be forbidden. Has to be forbidden. Some people are using it, but it has to be very restrictive. And you enter into a totally different world. It's a world where energy is provided and then is going to be used in another way and so on. So energy is provided. There is another point I would like to highlight, which is, and then I will show you a movie and I will ask you to meditate around this movie, is that I'm remaining at the moment at the scale of the molecule, which is the nanometer. And I want to remain on this for some time. If I have energy provided, and if I have my Brownian molecule, whichever that is, how am I going to recognize that it is a motor? For those of you who never saw a molecular motor, basically, I'm moving a little bit forward. It's if the definition of active matter is that I'm providing an energy and eventually I can transform this chemical energy into mechanical energy, I'm equipped to start building a molecular motor. And now I'm asking you the question, how would you recognize a molecular motor? It will have a directed movement, but I think both of you or many of you are already extremely high in the progression, so I think I agree with you. But those of you who never saw a molecular motor, what do you hear in the word molecular motor?
I can understand that
generating some kind of energy through which motor action,
More?
motor is working through that.
That's right. But how would you recognize? I mean, when you hear molecular, when you see molecular, it means what? Molecular is going, which scale is it going to be?
Size.
What is the size? Yes. What is the size? It's going to be nanometer, right? So we are in the nanometer. Now the question that I'm asking is that, okay, I'm defining that active matter is using energy, the chemical energy is going to be transformed into mechanical energy, and eventually I can provide something that is going to be a molecular motor. What does it's a motor, how would you recognize a molecular motor, right? If I take a particle and the particle is jiggling around like this, can I call that a motor or not? Why?
The pattern of the motion is very random.
The what?
The pattern of the motion is random.
Is random, right? But could I not imagine that I have a particle that is going to receive energy and transform it into mechanical energy that eventually will be also random, right? So how can I recognize a molecular motor, essentially? That's basically the point that I'm trying to make, and that's why I was calling this lecture of today active matter, living matter. I'm talking about molecular scale at the moment. Before 1 p.m., I will go to cells and cell motion, but I want to go very, very slowly here. And so basically, how can I recognize a motor in its own right? You don't know anything about the system, you would observe an object at the nanometer scale, and you would say, ah, this is a motor. Have you ever seen a molecular motor? In which context?
So it's organized form in one direction or active direction.
In one direction, so you go also in one direction. Anyone has a clue or not? Basically, when you think about a motor in the macroscopic world, probably you think like me of a motor like a bicycle, like a car, which is going to one point to another one. So let's dwell on this for a moment. And I'm going to say that a molecular motor is going to be a motor that is going to be directional motion. So it means that if I'm in a point A, I'm going to be able to move on the point B at the molecular scale. And in this simple word, actually, there is one of the key difference between passive matter and active or living matter. The fact that the living matter is having a breadth of different molecular motors is essential, as I will try to show you now in the next 15 or 20 minutes. Imagine the particles of yesterday, which were jiggling around. What would you need to do to make them move directionally? What do you say?
Energy.
You need to give energy, right? And how would you impose the direction?
We have to give force in some direction so that they will move that.
Okay, but what is the difference between the flow that you observed yesterday and the direction motion of a single object, right? If you tilt like this, it's easy. All of them are going to move like this. It's not what you want. What you want is that a single motor is going to do this, or another motor is going to do that.
That force is applied on the fluid. Meanwhile, here we need the force on the molecule.
That's right. That's the key difference. So here what you have is that you have a molecular motor, and it's a force that is going to be, you are almost in the word that is used in the community. Instead of having a global flow, what you want to have is a force that is local. I mean, that's what you were saying in effect, or spontaneously driven. Basically, the motor is going to hydrolyze the ATP to give a mechanical energy and eventually to move in one direction. So I need to break the symmetry. There's going to be something on the right, and there's going to be something on the left. So I'll show you an example of a movie that you could find inside the cytoplasm. I could multiply the movies. You can go on YouTube or Dailymotion, you name it, and you type molecular motors, you will see this type of motion all over the place in living matter. This is really a very key fundamental property of living matter. So this is, again, one of the many movies that you could find on the internet. And we're going to see this movie sometime, right? And I will ask you questions, and then we'll turn to the blackboard. And at the half an hour later, I will show you another movie. So this is taken inside the cell, and this is what it looks like. You see the time? It's real time. What do you see? So raise your hand, and I would like to hear as many women as men. Let's start by the women, if I may.
Particles are moving in a particular direction.
All particles are moving in a particular direction, or some of them?
No,
Some of them, right. So how would you qualify this motion?
some are directional and some are non-directional.
Which one are not directional? I mean, you can be, I mean, what is striking, whenever you see that, you have the impression to be on what? I mean, you know, you can use everyday's word, right? It's not, doesn't have to be educated, kind of observation. When you see that, you think about what?
Traffic.
Yeah.
I can see.
Yes?
I know these things, so shall I?
No. Traffic. You think about traffic essentially. You think about highways, right? And you think about cars moving. But this is low Reynolds number, right? So there is no inertia. And what you see is that if you follow one track, if I follow one vesicle here, the vesicle is going to move here, and eventually, you know, it will cross, it will change, and then it will go back here. It will continue to move, and then there is another one coming at the back. When I have a track, are all the particles moving in the same directions or not? Yes? Who says yes? Who says no? Yes, massive yes. Look at the movie very, very carefully. You will see that on some tracks, look at this one. You see the particles? They were moving essentially in opposite direction. So it's like a road where there will be two roads. But the problem is that here you see the scale. The scale is 5 micrometers, so meaning that the road that we are talking about here are about 100 nanometer. So it's a 100 nanometer road, out of which you have one vesicle moving like this and another vesicle passing by, in the opposite direction. It's a good molecular motor, right? I mean, energy, I don't know, but what I'm sure about is that it's a directional motion. So there was one person defending Brownian motion. I think it was you. You were right. Where is Brownian motion here? I see you jingling your finger, but it's good, but where is it? It's difficult to, but you can see the Brownian motion here. Where do you see it? Yes?
Beyond the track.
Beyond the track, yes. If you go away from the track, you see some particles, which are going very, subreptitiously. You see some small spots doing like this. There is another way to see when the Brownian motion is, but it's extremely subtle. And I won't be able to mimic it with my own body, but you see here, this one moving around is Brownian motion typically. But when you follow very carefully one track, you see that some of the vesicles are moving straight, and at some point, they're going to go away from the track. Yes. And when they go away from the track, what happens to them? Brownian motion. It was we saw yesterday, so that's why I was insisting to distinguish the lecture of yesterday, which was inert matter. So whenever you have a vesicle with a particle, which is connected to a molecular motor, if it's in the cytoplasm, which is the name of the structure we see here, it's going to wander around everywhere. So if I am a molecular motor, I'm moving like this. I have a track on which I'm moving. And as soon as I'm away from the track, I cannot do it obviously because gravity is driving me down, but then I will go wandering in the room all over. Okay? So what we observe here, I will let you the privilege to say what it is because you know.
So, as far as I'm a physicist, but I have worked on molecular motors for some time. So these are what I feel is that microtubules
Yes.
on which there are molecular motors which are moving.
That's right. So these are microtubules which are in the cell. They are part of the 3 elements of the cytoskeleton of all the cells. Actually, you will find them in plants, in yeast, budding yeast, or yeast of the bakery, or yeast of the beer. You will find them all over. You will find them in Drosophila, in the fly, in zebrafish, all over. What you see here is that you have, sorry, microtubule, a molecular motor that is either kinesin or dynein, and I will write them on the board so that you know them. And they move on this track, and it is the interaction between the molecular motor and the track that makes the motor be operational. And they have the quality also to hydrolyze ATP. So somehow they are eligible to be good molecular motors. So this is one example, if you wish, of local molecular motor. You see the difference between what we see and what you saw yesterday. You had Brownian particle all over with a global flow and all the particles moving because your sample was tilted. Here, somehow you will see that you have motion of all types. There is no global flow, and the particles are moving, you know, very regularly with almost a constant speed on the track. Last question on this movie, and then I will close the movie. According to you, what is the gain of a cell to have these tracks? When you have a biological phenomena, apart from the new physics, out of equilibrium physics, it's important that you ask yourself the question, what does it serve for? Sometimes it serves nothing, right? It can be, but usually in terms of evolutionary understanding, usually there is a function, otherwise it would have disappeared. This is an hypothesis somehow. But how useful can it be to have a directional transport inside the cell?
Efficient transport of cargo within cell.
Yes, efficient transport of cargo. Because if there is a damage of a cell somewhere, if there is a cortex that is broken somewhere that is to be repaired, it's better to be directional than diffusion. Because as I said yesterday, if you want to go from here to Bangalore, going straight is the best way. Traveling around throughout the world is not very efficient, right? So basically, you'd rather, right to the point, having a microtubule going from one spot to the other to eventually reach that spot. Can you give me a back on the envelope of velocity? Because you have the time, you have the scale. We need that this afternoon. You will measure the velocity this afternoon. So what is back on the envelope velocity here? 5 micrometer. This is real time. How long does it take to make 5 micrometer?
5 minutes.
5 what?
5 minutes.
This is hour, minute, seconds.
Oh, that's
Second, yeah. So 5 seconds, 5 micrometer in 5 seconds. It's about 1 micrometer per second. Is it quick? It's not quick. It's not bad, right? 1 micrometer per second. If you think about the scale of a cell that we talked about yesterday, it's extremely fast, right? Low Reynolds number, but extremely fast. And so back on the envelope, I'm giving you this because we talk about chemical energy into mechanical energy. Do you know the gain of typical molecular motors in biology, or this type of motor? What is the efficiency? You give a given chemical energy, which is a number, 10 kT. You're going to have a mechanical energy. Try to guess what is the efficiency.
Very high.
Very high. High like what? 50%? What is the efficiency of the car?
20%.
I think you're optimistic. Molecular motor efficiency is close to 98%. The efficiency of molecular motors in biology are amazing, are simply amazing somehow, and we do not compete with them. There is no, at this stage, there is no way. They are extremely efficient. Good. So I close this movie, and I'll return to another movie later on. But you see that it's not, yeah, please.
Reason for this high efficiency?
I'm sorry?
Reason for it, that high efficiency?
I don't know. The true answer is that I don't know. Finding reasons of why a system is what it is a difficult question because it looks like epistemology rather than absolute argument. There's a counter argument, but there might be, but I don't know the answer, to be quite honest. So I'm returning to this, actually. Molecular motor. And as I said, if you have, yeah, please. There was somebody, no? The more the better, right? It's okay? So the molecular motor is going to be this object, which is nanometer scale, which is going to move directionally, 10 kT energy provided, 10 kT like this, and then If I'm going away, I'm having Brownian motion. Now, as you saw, I was distinguishing a biopolymer and what I was calling molecular motor. I'm going to represent the molecular motor like this. But the one I want to highlight is that it's a pair of objects. I need to have a track and I need to have a motor on top of it. You could see, I mean, these were the microtubule that were below. If there were no track, it would not be working. So now you get used to scaling arguments. Energy of a particle is kT, energy of the molecular motor is going to be 10 kT. Try to guess what is the energy of interaction between the motor and the biopolymer. Again, think about energy like Feynman, it's a bank account. You compare how much you put and how much you take out, and you compare orders of magnitude. So, energy of interaction between the motor and the biopolymer, can it be kT? No. 10 kT?
About between 1 to 10.
1 to 10?
Yeah, in between.
It's, yeah, 1 to 10 or even a little bit more, right? So let's say that 10 or 20 kT-ish. So the interaction between these two objects is going to be about 10 kT because you want to compete with the molecular motor that needs to detach at some point to eventually go somewhere else while the other one is attached, because if it is detached, it will go in the air, and then it will go this again. So we have now our good molecular motor. What I would like to show you now is that it's an essential property of active matter and living matter. It's not an epiphenomenon. It is essential. It is key. It is everywhere in biology. And I'm going to list out a certain amount of molecular motors that have this key property. So for this, I'm going to go through a table. The first element is going to list out the biopolymer. So biopolymer is here, and I'm going to name them one after the other. Biopolymer. The first one is, I'm going to start by the microtubule. I'm going to go the other way around. I will start by the DNA. And I would like that you name for me the molecular motor. Do you know a motor associated with DNA? DNA polymerase, RNA polymerase, etc., etc. Helicase, topoisomerase. Basically, you have a variety of protein that will have a function which is going to work on this DNA. What is the function of DNA polymerase? What does it serve for? Yes? Yeah. DNA polymerase is going to take the DNA somehow and is going to transform it into a new DNA. Right? So you have the molecular motor that is here, you have the biopolymer that is right here, and then basically I would like that now we give a function with a plural. So DNA polymerase, what is a function?
DNA replication.
Yes, but more than yeah. So basically replication. What else? So replication as a whole. The topoisomerase is going to be which function? You saw them in cancer. For those of you who worked, yes. Topoisomerase. What is the? Yes, if there is a calling, the topoisomerase is going to come and to cut, and then allow, relaxation of the stress, and eventually recall it. So basically, it's a series of enzyme, which usually finishes with an ACE, which are going to have a function about repair. I'm going now to another biopolymer. I'm going to the actin filament. Do you know where they are? Actin filaments. For those of you who made biology in high school or even before, muscles. What is the molecular motor that is associated with actin? Myosin. Do you know which myosin is associated to it? Myosin. Okay, the ones who are biologists, you learned that in your curriculum. It's myosin 2, right? But it's a superfamily. What is the function of actin and myosin? To contract, and in general, it is using for what? Yesterday I was talking about, embryogenesis, when you start from a cortex, a cell that is proliferating, eventually dividing to give an organ. What is the function of actin and myosin? All these processes is to do what? To contract, but in the muscle, it's muscle contraction like this. In the context of the embryo, it is simply to shape the embryo. So basically, it is the assembly which is necessary to shape or to sculpt cells and tissues. And the last one that we just saw a second ago is a microtubule. The two motors that I mentioned is kinesin and dynein. And what is the function of it? Cargo, it's going to be directed transport. I want to mention, basically this is a bit a catalog, but just to make you have the feeling that somehow wherever you go inside the cell, whatever species you are considering, in the spatial scale I was showing yesterday, you will find them all over. You find them in the nucleus, you find them in the cytoplasm and everywhere. There are other examples that you could think about. Maybe it will come to you when you... Other molecular motors that you have stumbled upon necessarily during your curriculum, yes?
The one which creates generates...
Yes, so this is the one I wanted to mention. So I put it with the second line here. It is the F1 ATPase. That's the one I think that you had in mind. So it's F1 ATPase. I will say one word about it. It's going to be involved into ATP generation. So the one that you are mentioning at the moment is an extraordinary success of structural biology. There was a famous scientist whose name is Walker, who had made the crystal structure of F1 ATPase. And he observed it, and he said, it has a shape which is extremely symmetrical. It looks like a rotor and a stator. So he looked at it and he said, I attended his talk, in fact, it was in 95 or 96. That's the way he was making the storytelling, actually. He observed this beautiful crystal structure and he said, it has this symmetry, but in evolution, this is what happened. It used to be in the back of a flagellum motor of a moving bacteria. And then this flagellum motor and this bacteria, once upon a time, entered into a cell and eventually lost its bacteria, and it became a mitochondria. So probably, if the mitochondria is the result of an ancient bacteria, to generate ATP, it would be interesting to consider that the difference in potential across the membrane of mitochondria could give the energy to allow the rotation of F1 ATPase to make the ATP hydrolysis possible. And he was right. He got the Nobel Prize for it. So basically, it was not it's a life story somehow. And you might see on internet a very good experiment, amazing, by Noji, who was in Japan, who is still in Japan, in the same lab, where he took the F1 ATPase, he grafted an actin filament, and he could see by grafting the actin filament was rotating in the presence of ATP, showing that indeed it's a molecular motor. I'm going fast on it because I hadn't prepared to talk about it during this lecture, but just bear in mind that this is another example. Continue the example. The most biologists in the audience, what other molecular motors you can think about? One thing maybe to guide you so that you have it, the intuition very quick, the molecular motor, the main readout to see it is directional motion. If you see a directional motion at the level of a molecule, it is a molecular motor, basically. It's not, it can be, it is. Why? Because otherwise KT is randomizing everything. So if you have a directed motion, it is a molecular motor. So maybe one last example about molecular motor. Yes? The channel? That's a good one, yeah. That's one. The one that I had in mind was more in the translation. How does the translation operate? Transcription, we talked about it. That translation goes from? Ribosome. Yeah, ribosome. So what does it happen? You have the conversion between the RNA, which is then eventually to be converted into a sequence amino acid, which eventually is going to be peptide, and that's going to be eventually a protein. That's another example of molecular motor. So far, so good? So you can have replication, repair for the genome, which is the molecular scale I was mentioning yesterday, which is meaningful in terms of function. You have shaping of a cell, mitosis, separation of two cells is going to be through actomyosin also. Motion, that we're going to see this afternoon and to some extent also tomorrow, is going to be mediated also by actin myosin filament. Microtubules are going to allow transport and to escape diffusion inside the cell. F1 ATPase to generate ATP. They have in common that it's a directed motion. Let's meditate around this. I managed to read Global Initiative on Academic Network course because of what? Why do I manage to read that?
Because you can see.
Because I can see, that's first point, agree with you. What? Yes, and what? Because if it would be written in Hindi, I wouldn't be able to read it because I don't know yet Hindi, but you would be able to read it, right? But the point is that beyond this knowledge, why can I guess that it is written global initiative?
Because of the light falling, right?
Because of the light falling, yes, that's right. But in addition to what's written here, why can I read it? Okay, imagine, let's go like this. ESROUCKROWTENCIM. I won't be able to read it. It is organized. There is another word that I would like to hear. It is
Patterns.
patterns, ordered. I'm not talking about it, not by accident. It is, yes, a word that I keep using for an hour now. It is, what? Directional. Essentially, it is directional. In Hindi, from which direction to which direction do you? It is the same one. So basically, you go from left to right, and somehow a text can be understood and can be essentially read because there is a direction, essentially. So think about it, that somehow you can make sense of a text because of its directionality. It is exactly the same for the DNA polymerase or the RNA polymerase. You have a polymerase that is going to enter into a double-stranded DNA and eventually to be moving directionally to eventually read the sequences one after the other. For single molecule experiments, and we won't cover it in this lecture, that would be too long, but basically what can happen is that during the reading, there is still Brownian motion. So eventually, the particles might move backward and eventually continue forward and make an error and then move backward. But eventually, it will move in one direction. And that's how this directionality is absolutely essential. The same way we can read this text, the same way the molecular motors are going to operate. The same I applied for stress generation. If I have stress generation, here basically it's going to be the reading of a polymerase, which is going to be directional. For stress generation, if I'm taking the muscle, I'm depicting here the actin filament and the muscles. There is a polarity here of the actin filament, meaning that there is a front and there is a back. And because the motor interacts with this particular interaction and direction, eventually it will contract. The same for the microtubules and the kinesis. So the idea of a directional motion is absolutely essential. It allows to read, it allows to apply a stress, it allows to have a transport that will be directional from a point A to a point B. So directionality is absolutely key in all this story. I'm going to wrap up this part and then I'll move to some other parts, just conceptual. You can see that this is a generic property of description of molecular motors. It's an essential property of living and active matter. We find them all over. A way to recognize them is simply to observe. If I have an object moving directionally, it is a good molecular motor. This is eligible for being a molecular motor. The energy source is most of the time ATP, or ATP hydrolysis to be more specific. And for the DNA polymerase, or for the RNA polymerase, it's going to be the NTP, ATCG that I was mentioning before, which are going to enter into the replication wheel. Okay? 10 kT is larger than kT, so it is outcompeting the potential noise that will be all around. One category that I would like to introduce now is the duty ratio. Duty ratio is a ratio, so it has no unit. So essentially, I don't need to basically think about what type of physical realm it's going to correspond to, but I'm going to need it in terms of understanding of how molecular motor operates. The duty ratio is the time of interaction of the biopolymer with the track over the time of hydrolysis of ATP. So it's a ratio. It's a ratio between two times. And you're going to see that it's extremely convenient to evaluate the way the different motors operate. It's extremely efficient. So I will let you to try to guess what can be the meaning of the duty ratio. Let's start by the microtubule here. Microtubules are this one. Maybe I'm going to go here. Microtubule kinesin duty ratio. Try to think about it. Is it a number that is going to be high or a number that is going to be low? I am the motor. I am the motor. I'm moving like this. During that time, each time I have a motion, I'm hydrolyzing ATP. There is a background periodic noise. It's okay? Good, it stopped. So I'm moving like this. The duty ratio is going to be what? What do you say? Greater than 1? Well, the time of interaction of the biopolymer on the interaction of the motor on the biopolymer, total hydrolysis of ATP, you know, let's say this is one motor. I'm hydrolyzing ATP. I'm moving like this, and then I'm going to bind again. The duty ratio, you said it almost right, but it's a bit wrong. The duty ratio has to be less than 1. Okay, so it's going to be like 0.3 or it's going to be 0.9? Who said 0.9? Yeah, 0.9. So you say it's going to be like, 90% or it's going to be like 90%. Why do you say that? It's correct. I mean, it is the right guess. What's happening? I have my foot that is behind here, and I'm going to have like 90% of the time I'm going to be bound to it, right? But 10% of the time, it's even larger actually from microtubule kinesin, is that somehow I'm going to be able to move a little bit, because if I don't move, I will never eventually be able to find the next track. So it's more like it's superior to 90%. Now, if I'm going to the other example, which is going to be this guy here, the actin filament. For the actin filament and the myosin, try to guess what is the duty ratio like. So think about it, right? I'm going to make you, an image so that you see where I want to go. This is a typical muscle where you have a sarcomere, and you have here the myosin, which are interacting like this with the actin filament, which are in this color here. And so basically, what do you expect or what do you want to have for the duty ratio in that particular context? What do you say? Greater than 1? how can you have a time of interaction of the motor to the biopolymer track that can be larger than the time of interaction of ATP? This is the total time of hydrolysis of ATP. So I'm going to do it with my hands. This is the myosin head bound to the actin filament. ATP arrives, the myosin wander around, eventually change conformation, reattach to the actin filament, and gives a kick. So it means that the total time that is the longer is necessarily the time of hydrolysis, right? Is it okay? So then the time that I expect here has to be, maybe to be more fair, I should represent it a little bit differently. I'm going to add some more motor here so that you get a sense of where I want to go. If I have many, many motors acting in concert, what is the duty ratio that is going to be meaningful so that it can be working? You probably know this game where there is a rope and you are, let's say, half of the room is having the rope and pull in this direction, and you, half of the rope, are having a rope and pulling in the opposite direction. What is the main strategy so that you will be helping each other so that one of the two teams will win and will pull in the one direction? What is the main, one of the strategies that is working? Yes? Yes?
Pull at the same time.
Pull at the same time, but obviously you won't pull at the same time because some people are cooperative, others are less, you know, they're more individualistic. And, what do you say?
Coordinated effort.
Coordinated effort. That, I agree with you, coordinated effort. But the thing is that you cannot command one motor to command the next motor as human being, right? So basically, if they don't want, they don't want. It won't happen. So basically, what is the strategy so that it's successful, that eventually is going to work? Yes? I'm talking about duty ratio, so I'm trying to make a sense that somehow the duty ratio would rather be 50? 3%. It's not necessarily intuitive somehow, but it means that if you need to generate a stress, it's better that somehow you give a small kick, and once you give a small kick, after that, you go away so that if there is a kick by another molecule somehow, you do not impede the action of the other one. I'll give you an example. There is one motor here attached, another motor here attached, there is one kick here. If this guy is attached too long with the polymer here, it won't do anything because then it will essentially be blocked. So imagine the metaphor of the rope, it will mean that you're pulling on the rope and the person in front of you is not moving. So basically, you're pulling like this and the person is right here is like this, right? You would be nervous because somehow what you are doing is destroyed by the guy who is essentially right nearby you. So the strategy is just to give a small kick and then, you know, go out. The next beat would be a small kick, go out. You need to be attached, but eventually it would be working. So I'm presenting this because somehow, if you have the energy source of your motor, if you have the polymer that is associated to it, if you have the duty ratio, you are pretty much equipped to start thinking about your molecular motor of interest. Usually these are the basics which are absolutely indispensable. So all these molecular motors have gained a lot of interest since the 90s. As you probably know, there were some synthetic motors that were produced, different Nobel Prizes that were given, and I have to say that one of the recipients of the Nobel Prize for the molecular motors was from Strasbourg University, my university, Jean-Pierre Sauvage, for the synthesis of the rotaxane. So this is a synthetic molecular motor. For the chemist in the room, probably you've heard about the molecular motor, and basically there were 3 Nobel Prize recipients, one of them passed away recently. But it's all around this concept that biology is offering a variety of molecular motors that are extremely efficient, and synthetic chemistry should be able also to have molecular motors that could drive also new material. But the concepts are exactly the same. It's low Reynolds number, an energy source that you need to provide, a polymer and a track, or basically 2 objects in interaction, time of interaction during the total time of the cycle, which is duty ratio. You will retrieve all the reasoning that are needed to build molecular motors. So to finish this part and then go to the cellular level, I want to give you a last hint about motility assay, which have been designed essentially to perform this type of experiment, but now out of the cell. So all I talked about so far was at the cellular level, and just a way of saying that you need these molecular motors to eventually transform inert matter into active matter. Now, what I want to show you now is a motility assay. They were designed some time ago, but they are extremely elegant experiments where you can purify the protein and reproduce molecular motor action. So the way they do that is that they purify it and they put on the surface myosin. For instance, it could be kinesin microtubule, it can be any protein that you think about. And on top of that, they come with the polymer. Basically, as you remember, it's always a pair of a motor and the biopolymer. So they come with a polymer on top of it. And they put, of course, an energy source that is ATP. That's it. So it's a kind of reductionist approach where you consider essentially the molecular motor, the track, and you let it go and you see what's happening. Anyone has seen this type of movie, motility assays? Yes? 1, 2? That's it? Right. So I'm going to show them to you and we're going to meditate on this movie, right? I mean, I invite you whenever you see the movie to try to observe everything you can see on the image and to try to put words in what you see. So this is going to come next. So now I'm repeating, I'm out of the cell, the proteins are purified, and then the only thing you try to do is essentially to observe and to try to make sense of the observation. Yes. I need to press this guy. So what you see in fluorescence is the actin filament. I could reproduce the very same movie with any of the motor that we talked about, or the biopolymer. It would be a bit different, but it would look like the same. So hopefully the image is going to come, and now you're going to see. So before, the first movie was essentially inside the cytoplasm. This one is an in vitro experiment. That's the word that is taken for this type of experiments. And then you're ready to go in a second. Yes, I need to plug in. Yeah, that's right. Thank you. Here we go. Each filament is 10 micrometer long. It was working a second ago, so it's going to work again, hopefully. Okay, now it's working on my computer, so probably it's going to work. I'm going to show it on the screen. One more time. This is real-time observation. What do you see? Just with words. Use your words. I think your words are very good. You don't need to have educated guess. It's really like, what do you see?
Worms.
Worms, right? It looks like worms. You have the impression to see worms, right? I'm repeating, these are biopolymer, right? So, well, so, yeah?
Some elongated objectives.
Some are?
Elongated.
Elongated, yes. They have a finite length, but eventually, yes, you are absolutely right. They are elongated and they look like worms. What makes you feel that they are worms-like? There is something that seems like a worm is much larger, right? We're talking here about a nanometer, 7 nanometer thick biopolymer and 10 micrometer long. So basically, what do you see here? Are they moving all in the same direction? No, obviously they're not moving in the same direction. Some are moving to the right, others are moving to the left. So basically, there is something weird here. Yes?
So it's looking like a bacterial.
It looks like a bacteria. E. coli, right. So we'll come back to that in about 10 minutes about the cell here at the moment. But I'm insisting on this here, it's only a biopolymer. So E. coli, 1 micrometer here, 10 nanometer, we are really scaled down. Right, so what else do you see here in the movie, which is kind of interesting? Whenever you have a filament, is it moving, is it changing direction? Yes, it's changing direction, good. Now, whenever there is a front, is the front remaining the front or not? Right, so it means that if there is a front and a back, you can follow the movie, and basically, you can follow it again and again and again. You can view it 1,000 times. The front here remains the front. It's strange. The front remains the front. It can change direction. It can go all over the place. But I can tell you, if you suppress the energy source, if you suppress ATP, there is no motion. So you need ATP for the motion to be. So what I was writing here is true. Basically, you need the energy source to eventually get the motion. There is some funny events. I don't know if you could spot them. Sometime, maybe some of you are going to visualize it. Yes, please. No, the more the better. Yeah. I mean, at some moment, you're going to see something very strange. There are some, when you say, yeah, there is some kind of rotary guys here. I mean, there are some points, what's going on there? These are the same filaments as all the other filaments with molecular motor. How come there is here a rotation that is showing up here? What do you say?
Myosin that are there has to do something with that.
Yes, that there is something, you have many motors that, the metaphor of a cord and many people pulling down, many motors pushing like this, and might be a defect somewhere where a molecular motor is not working. So eventually a defect might show up at that point. I'm fascinated by this movie because the question that I want to ask you is, how can it work? Right, you prepare the sample, you have molecular motors that are bound to the sample, you come with an actin filament at the top, right? And then they're going to dwell on the surface. So how eventually are you going to get a directed motion? Because the motion is directed. Everything is good here, right? We have all the elements that are gathered that it's working. How can it be working? Yes, so we are all like this, right? So basically, yes. It's doing like this, but I'm a motor. How can I know that I should push in this direction?
There may be some kind of asymmetry in the structure.
Chemical, so you talk about asymmetry, so this is very important. What did you say?
There could be some force which attract chemical things.
Yes, but the point is that everything is isotropic here. So basically, there cannot be any source of chemical anisotropy. Essentially,
The motors are uniformly on the.
they are totally homogeneous on the surface. So there is something which is extremely strange that it's working. I mean, I'm repeating. You have molecular motors on the surface. You come with filament at the top, and the filament at the top, essentially what they do, they're going to interact with the surface. The myosin motors are going to give some kicks. And for some reason, all the motors are moving directionally. All the filaments are moving directionally. It's not one sometime and the other one is not. All of them are moving. Sometimes what is funny in this movie is that you see here, this is a defect that you can see here. Sometimes the filaments are in the surface and they go away. I don't know if you can see that. You see some small filaments that are going here and then they disappear. What happened to them? Yes?
They move up to the focusing area.
And what happened to them when they escape to the surface, escape from the surface?
Fusion.
There is? Diffusion, right. KT is back. So basically here we are competing with KT. It's going to be 10 KT interaction with the molecular motor. So the point that I'm trying to highlight here is that it's very strange that it's working. And it is the last point I want to highlight on the molecular motor is that there is an inherent property that is breaking the symmetry, and I would like you to tell me which one. How come it's working? How come all the filaments are moving? I'm repeating, imagine you design an experiment from scratch, you and I would not be able to make it unless there is something that I'm introducing, which is key actually to the molecular motor's action. Any guess? Can you switch on the light, please? Thank you. Yes?
Is there only one motor under that microtubule? Or
So this is here, what I showed to you here, the one that I showed to you was actin filament and myosin motor. So we were really in the context of low duty ratio, and it was an ensemble of myosin motor interacting with actin filament. So it was very different. If I would draw here, to be fair, it would be like more in 1D, it would be many, many myosin motor interacting with one actin filament. So I would like you to guess actually what is the source of broken symmetry. How is it working? I'm sure you know, but I just would like you to guess what it is. How come you have a direction? Again, the experimentalist, what does the experimentalist prepare? The experimentalist prepares a surface like this with myosin motor. And then he comes at the top with filaments that are all around. The filaments eventually are going to arrive on the surface, and when they arrive on the surface, eventually they're going to move. How come they all move in one direction? Yes, I would like that I don't say it, but you try to guess it. Swimming? I heard swimming?
Yeah.
So the short answer is no, but it's interesting that you say so. Swimming, it means that it is in water. Here, you have only diffusion, and eventually, when you are bound to the surface, you're going to crawl, basically. It's crawling that's going to happen. But why is it directional? So the short answer that I'm going to give at the moment, which is the last element I wanted to bring about the molecular motor, is that all these biopolymer, all of them, they have a chirality. What is chirality? Yes, they have a pitch. There is a direction. The symmetry is broken. So somehow, when you think about the DNA, the most biologists, what is the DNA structure? Double helix, which is going to have a pitch. What is the characteristic of actin filament? It's also a double helix with a pitch. What is the characteristic of microtubule? It's also a helical structure with a pitch, etc., etc. So all these structures that I mentioned so far have all a broken symmetry, all of them. It's not that they might, it's that all the ones that I mentioned so far have a broken symmetry. So you see that now there is the introduction of an important concept here, is that the broken symmetry that I was talking about, the directional motion, is tightly associated with the chirality of the biopolymer, coupled to the interaction with the molecular motor. This is very easy to say. It's more difficult to digest somehow, because it means that everything is encoded inside the structure of the biopolymer themselves, which are working the symmetry. It is absolutely essential. If you don't have a broken symmetry, the molecular motors will not have a rule of interaction. So if you try to recapitulate what I was saying so far, essentially what's going on is that you have, returning to microtubule kinesin, these are the 2 feet of the kinesin. The molecular motor is like here, is detaching, hydrolyzing ATP, and then looking for another binding site to bind. Eventually it will bind, but why does it know that it should bind here versus here? It is because the chirality of the microtubule is present together with interaction with the motor, actually. So that's why I'm saying the chirality is essential for the broken symmetry.
So the track and the motor both chiral.
Both are chiral, but it's first I mean, why I'm insisting so much on this is that in the motility assay experiment, how come the orange filaments could be directional? The only explanation, as far as I know, is that the filaments are chiral. Because they are chiral, if I am a motor on the surface, I'm going to twist or to do anything possible to be able to bind to the actin filaments and then give the kick in the right direction. In the next motor, I will do the same. I will do a kick and so on and so forth. So basically, to be honest, if I want to show them in the right way, I should draw them systematically with the chirality embedded so that the motor can twist and then have the interaction. Otherwise, it could not be working, essentially. It could not be working. So I think you get a sense of why I'm so focused into the broken symmetry, directional motion, in terms of function for the biology, for transcription, for translation, for stress generation, for transport. It's an obsession because living matter is essentially an assembly of molecular motors which are outcompeting diffusion to eventually lead to biological function which are as essential as genome integrity, cytokinesis, shaping embryo, transport in the cytoplasm, and so on. This is the building block. I saw two questions. I think I saw some, yes. Do you were first and second, yes. You need to shout because I don't hear. Yes.
These molecular motors
Yes.
rotate directionally.
Yes.
My question is whether they are rotating also.
So that's a very good question, really good. So the short answer is that they do. Some of them do. And in fact, I haven't scheduled to talk about it, but if there is a massive interest on it, I can shift a bit the lecture of the days which are going to follow. You can make single molecule experiment with this molecular motor and optical tweezers, actually. And if you take the polymer in suspension, you can observe, if you are a good experimentalist, the rotation of some of the motor around the helical pitch, and you can reveal the pitch. So yes, the short answer is that they can. And it goes together with the pitch, because somehow you can reveal the pitch by this type of experiment. That answers your point, I guess.
When we say symmetry is broken, actually, what does it mean?
What does it mean? That there is a front and a back. In a helical pitch, if I have a helical pitch, it means that if I'm going in this direction, I'm breaking the symmetry because now I have a direction. I'm rotating in this direction and I'm not rotating in the opposite direction. It's if you wish, a qualitative statement about broken symmetry is chirality. The space is not anymore isotropic. There is one direction that is going to be favored. And it is at the core, actually, of the active matter, living matter, is that the symmetry is broken at every molecular scale. There is always a broken symmetry that is encoded inside the chirality or the interaction between the molecular motor and the track. That's why I wanted this lecture to be day 2, because for this, you need the day 1, where instead you take the inert matter as being isotropic, at equilibrium, and everything is equivalent in every direction of the space. This is the background. We evolve out of this. Now, active matter and living matter, you add an energy source, you break symmetry by directionality, you introduce chirality, and once you have all these components, you can start having all these essential functions that are indispensable for having a functional cell. You see the progression. I mean, you see what I mean. Yeah. If you want to have an idea of broken symmetry, again, it doesn't have to be very sophisticated word. It's essentially broken symmetry. It means that front and back. The filaments that were moving like worms, there was a front and there was a back. So I think second and third, yes.
So, it's not related to this. If you imagine, a zygote is spherical.
Yes.
And it's symmetric.
Yes.
From a zygote, you develop an organism with a head and a tail.
Yes.
So does chirality also have a role to play there?
So it's a very important point. I mean, I'm going to say it very briefly now, and I'll be more elaborated, I hope, later on in the week. Here, I am voluntarily at the molecular scale, and I'm going very slowly at the molecular scale. The integration from the molecular scale to the mesoscopic scale, so when you go to 1 filament to 10 to the 3, 10 to the 5, 10 to the 6, 1 million filaments, is a challenging gap. And in one word, which would be very unclear, I'm afraid, but I'll still give it, is that if you have this chirality, it might have some similar consequences for the million of filaments assembled. but it might not, because it depends on how you organize the filament. So the passage from one filament to an ensemble of filaments interacting is a subtle one. It might be sufficient, but there are some cases where it's not. I'll elaborate a little bit more on this, hopefully tomorrow. Okay. There was here a question that was starting. Yes?
So it is like the molecular machines are tending to move in both directions, but due to asymmetry of the filament.
It's, so it's good that you asked the question. Both are necessary. Both are necessary. First, the chirality of the filament. Because again, maybe I will ask the question the other way around. You remember the movie with the filament moving like this, right? Imagine if the filament would not be chiral. The filament is floating in the medium. It's arriving on the surface. This is the molecular motor, right? It's a good molecular motor. So it's going like this. It's arriving like this. Now it's on the molecular motor. How does the molecular motor can interact in the proper way with the filament?
It has to move in both directions too.
Yes, but the point is that if it would not be chiral, the molecular motor would equally push in one direction or in the opposite direction. So basically, because it's chiral, it's going to interact here, and the molecular motor is going to bind preferentially to one side and to give a kick also in a given side. So it's not only the chirality of the filament that is essential, it's the chirality of the filament plus the interaction potential, but I think you are a minority of physicists in the audience, so I don't know how much. I have prepared a theory for the molecular motor with interaction potential and equation and master equation. I don't know if I should go through it because it's becoming a bit formal if I go through it. I'll discuss this afternoon one-to-one. If you want, I can present it tomorrow. But basically, if you go through this type of model, you need the chirality of the filament and also the chirality of the interaction of the motor with the filament. But for the motility assay to work here, you absolutely need the filament to be chiral. If they would not be chiral, you would have a filament here, this motor pushing in this direction, this motor pushing in the opposite direction, and the filament not moving at all. And ironically, all the filaments are moving. So it means that it's a system that is pretty robust. I mean, it allows to have and it's rather be robust because if it would not be robust, you would not have any of these phenomena. They would be forbidden because somehow it will sometimes work, sometimes not work. They have to be super robust to be working.
Sir, those molecules which are not moving in the living system, so that are not they are not symmetrical, but they are symmetrical.
So I'll be very cautious because if you see something with your eyes, with the cause can be multiple. And that's, you can have a situation where the interaction of energy is so strong that somewhere it cannot move. Another example, if there is a depletion of energy, if there is no ATP, the state of interaction between myosin motor and filament is, as you know, the rigor state. A rigor state, it means that the myosin is bound to the filament. It's not moving. So you see what I mean? Absence of motion has to be taken with due care. You cannot conclude immediately in the absence of chirality. It needs more, you see what I'm trying to say? It needs more elaboration. But I see your intuition. At the back of the envelope, as you probably know, most of the amino acids, most of the peptides, the symmetry is broken as a general rule in most of the biology, actually. So the broken symmetry seems to be more, there are some exceptions, intermediate filaments, there are some counterexamples about absence of broken symmetry, but they are pretty rare in the biological realm. So somehow, it seems like chirality has a fundamental function, and the point I'm trying to defend in the second lecture is that it is essential to transform inert matter into living matter through all these functions that are essential for life. There was another question starting here, yes.
What I used to think about the directionality, sir, there is this dissipation that takes place and there is a time reversal asymmetry.
Yes.
Here we are seeing that there is also a structural asymmetry.
Yes.
There in systems.
Yes.
So then how is this, I'm not very much, I'm not clearly understanding how this chirality is coming into the picture. But I was thinking there's some binding side where this motor binds to.
Yes,
and
yes. So you are, I think the point you are highlighting is that at the moment, I'm focusing essentially on the chirality of the filament, which is a structural chirality. And at the same time, and I think it goes to one of the questions that was asked here, it goes together with an ambiguity about defining a chirality of the interaction. And basically, you're telling me, could I specify which one is chiral? Is it the filament, or is it the interaction potential between the motor and the polymer? This is your question. Is it?
No, I'm saying that maybe both are there.
That
But,
both are there, that's the point.
I am not clearly understanding how is this chirality coming into the picture. I mean, the motors are chiral and also the track is chiral.
Yes.
And what is exactly happening?
Okay, so maybe I'll draw it here to make it more clear. What you have here is essentially motor which are deposited on the surface in every direction. And then comes a filament that is going to be having this chirality. Okay? As a matter of fact, what's going to happen next is going to interact with the motor. So I have one motor coming here. It's going to bind a very specific site. So we will find its specific site because of this chirality. And because it finds its binding site in the pre-particularity, it's going to then give a kick in this direction. Okay?
Based on the binding site.
Based on the binding site, which is mediated by the chirality of this guy. You see what I mean? Why I'm insisting on this? Because imagine if this actin filament would not be chiral, how the motility assay could be working? It would be impossible, because you would have filament that would come, some motor would push in one direction, other motors in opposite direction, and there is no way, all the filaments are moving. So it means that there is an intrinsic broken symmetry that is remarkably robust.
And how is the motor's chirality coming into the picture?
Because once they interact in the right side, they're going to also give a kick, a push, in the direction that will be always the same. So it's a dual broken symmetry, if you wish. It is the filament that is going to impose the direction, and it's going to be the kick that is going to be also in the right direction. So that's why it goes back to your question here, is that it's a dual broken symmetry that cooperate to eventually lead to the direction motion.
Just a hypothetical question. So if the energy of interaction
Yes.
is larger than the local minimum provided by this structure,
Yes.
if the energy of interaction is, let's say, an order of magnitude greater than that,
Yes.
then will we see a disappearance of this directed motion?
Yes, because if the interaction energy is larger, it won't be able to move. So that's, and I like that you say that, because somehow it allows me to justify, I mean, that was you who gave the answer earlier, but basically it is good that the energy is 10 kT, because if the energy of interaction would be 100 kT between the filament and the motor, you cannot compete with 100 kT, it's too strong, right? So if there is a binding which is 110 kT, 10 kT is peanuts. So basically you can give 10 kT, the motor will not see it, which is essentially the point you are making. So there is a kind of fine-tuning of the energy that is at play here, which is going to be quite essential. Okay. So far, so good. Any question? The simplest question, the better, right? And the more natural, the better. And then I'll introduce the lecture of this afternoon. Yes.
Yes, so sir, the energy which comes from the diffusion part, is it 100% suppressed? Wouldn't it be more efficient to use the energy of the diffusion in a particular time?
Excellent question. But I think you know the answer. What is the energy of diffusion?
kT,
What is the energy here of the molecular motor?
10 kT. Yes, so
Who is the stronger?
of course.
Yeah.
Yeah.
So it means that somehow the ATP hydrolysis, the energy source providing for the living matter, is 10 times higher than the Brownian motion, who eventually outcompete the Brownian motion. But somehow it's a fact, right? I mean, it's how it is. There is a famous chapter, again, for the sake of the week, I could not include it into the lectures, but there is a wonderful chapter, again, by Richard Feynman, that I talked about earlier for the energy, which is a chapter that is called Ratchet and Pull. He tries to imagine how a molecular motor could operate without, only with temperature. And he has a whole development I invite you to read the chapter because it's a fascinating chapter. And he says, okay, imagine if I would, can I build a molecular motor just with room temperature? After all, Brownian motion is a motion. So if I would imagine a machine where it would be ratchet and pole, where there would be a rotating vane, and there would be a asymmetric wheel on the other side, maybe Brownian motion will be able to jiggle this around and we will have a real molecular motor. That goes to your question, that somehow he's asking the question, can molecular motor exist only with temperature? And I won't give you the answer. I invite you to read the paper. For those of you who are the most physicists, he comes to the conclusion that maybe we need to say second principle of thermodynamics is wrong. Let's give up. Let's say that second principle of thermodynamics is wrong. And after all, you could say, well, maybe, okay, well, let's give it up. Maybe it's wrong, right? And that's the way his intellectual path is to come to a conclusion that I invite you to read. This being apart, Feynman being apart, what I'm talking about here is a 10 kT world. Yesterday was kT, today is 10 kT. And we are moving to 10 kT away from equilibrium, away from diffusion. It's okay. Other questions at the back, the one that I did not hear so far. Any questions, comments? Yes.
So in order to understand all this,
Yes.
looking at it from a broader, understanding. So is diffusion totally 100% suppressed in living systems?
No. That's a very, very good question again. Diffusion is not suppressed. Remember, whenever a transcription factor or whenever a molecule has to find its target, it is going to undergo diffusion. Diffusion is the background. All the molecules are diffusing around, or you cannot escape it, it's there. So you cannot escape it. It can have a function. It can allow, if you wish, accidental interaction that would never happen otherwise, right? Diffusion is good. I mean, just a side comment about it. Usually we live in an area where our interests are very focused in one direction. So this diffusion is becoming less and less possible, actually, in terms of human beings somehow. It's basically, we are focused, and more. This is a metaphor, obviously. But basically, diffusion allows to explore other space that you would not explore otherwise, and that allows, if you wish, a diversity somehow to be embedded. So diffusion is always there. Diffusion is also challenging the motor. If the motor is not attached, as I said, and as you saw in the movie, oops, it goes away and going to go somewhere else in the cell. So diffusion is a risk somehow. It could be also a chance in terms of differentiation. I'm shifting gears a little bit, because if the transcription factor are randomly finding the target in the gene, that would allow to have a variety of cell states. You know what I mean? So diffusion is there. It has a function. But for this, actually, you cannot challenge with diffusion. When you need to replicate DNA, you need to be precise at the base, right? It can't be having too much error. Otherwise, it can be lethal. If you need to shape the embryo, at some moment, you need to undergo gastrulation. Otherwise, no way it can work. If you have to have a transport for cytokinesis or for transport, it has to work. No way you can challenge it. So it's intermediate between diffusion allowing somehow homeostasis and variety of function with the noise being useful, sometimes, not always, but many times, and the other part, which is robust, key function of living matter. Good. I'm going to conclude in the last 10 minutes. One last question from somebody I never heard so far. I think I heard you. Yes.
Very important. I think
yes.
From my perspective,
yes.
Who takes the decision when to go from KT to 10 KT?
Nobody.
When's the, okay. So
I mean, the cell is a given, right? It just for a cell to be, it has to be. All what I described this morning about molecular motor, it is only one message. I mean, the key message of this morning is essentially to say the key property of active matter is molecular motor, and then diversity to guarantee essential function of living matter. This is the key message. And then KT, you cannot suppress it. It's here. But just you need to uncomplete it. There was one question here.
Is directly somehow connected to non-Markovic process?
Okay, it's an interesting point. Not necessarily. Because in the Markovian and non-Markovian process, you introduce the idea of a memory, essentially, is that the next step is going to be guided by the memory that you did before. Here, you can have some memory, if you wish, the polarity, the chirality of the filament is already keeping this memory, so it has some link, but it's not necessarily true. I could find counterexample where it's not working. That's why I would be very cautious on this extrapolation. I want to, yes?
Is there only ATP, a molecule which provides energy in molecular machines, or there any other?
So I mentioned ATP, I mentioned also TTP, CTP, GTP. Remember, for the polymerase, you need actually all these actually to generate all the bases of DNA. So basically, these are the other sources. Yeah. There was another, yes?
Could we say that directionality is the distinction between active matter and living matter?
I like that.
Because,
Between who and who?
because
Between
active matter and living matter.
no, this I don't want. between inert matter, on the one hand, the world of yesterday, and the world of today, which is active matter / living matter, right? So this is because inert matter is kT, active matter is 10 kT, and the issue between active matter and living matter, interrogation mark, and we'll discuss, yes.
So when you give example like bubble also divides and cell also
Yes.
divides.
Yes.
So in this case, directionality matters because bubble can divide anyway, but if cell is not divided in a particular direction.
So, okay. I like the argument, I would challenge it. If you have a cell that is undergoing, it increases in volume and then going to divide in the center, it could be reproduced by a subbubble. If you have a subbubble that is dividing in the center, it would be working. You see what I mean? So I like your direction, but I think this particular example would not work all the way through. Okay. Right. So in the last 10-ish minutes, I mean, if you have more questions, it's more important than what I want to say next. It's okay? So what we saw this morning, I will summarize it in 2 minutes before the end. Before that, I want to say, so what are we going to do this afternoon? This afternoon, thanks to Pramod, thank you, Pramod, Pramod has been taking some pond water. So it's pond water. Some of you have seen it yesterday, I guess, because you made some experiment with it. But basically, this afternoon, we're going to go back with the full scope, with the microscope, take your cell phone, take your computer. And now we're going to do essentially the exercise of taking active living matter in contrast to the inert matter of yesterday. And so the game is going to prepare the sample with pond water that was taken on campus. There are 4 sources. So you know the campus better than me. I arrived on Sunday evening, so you know where to find it. And so basically the idea is that we're going to prepare the sample, and I would like you to neutrally make the measurements or make the acquisition of what you see. Be as neutral as possible, right? You see what you see, you compute, first of all, you acquire with your cell phone long movie. It would be ideal that we prepare, if we manage 4 samples of the 4 sources, so that we have a sense of the comparison between them, and you will see what you will see. Try to acquire the movie at the best resolution that you can. Because if you manage to zoom, to focus very carefully, to have very, very large, basically a field where you can really see something, right, that is very clear and very clean, the better. I invite you also to play. You can take your sample, tilt it, see if it does something. You saw yesterday that it was doing something on the particle. You can see this afternoon if what you see in the pond water, it will do something, okay? So obviously this afternoon, you won't see the filament, right? This afternoon, we're going to go back to the cells. What is the scale of the cell? Micrometer, right? So this afternoon we'll be in the world of 1 micrometer, even larger for small of the, probably what you're going to see, but 1 micrometer. The world of this morning was the world of the nanometer. It's the spatial scale. So really, I'm going from nanometer to 1 micrometer here. So this afternoon, you will observe that. But it returns to actually one of the questions that you asked right before. Nevertheless, the principle that I described this morning will still be to some extent relevant to what you will see this afternoon. What you're going to see this afternoon is living matter, obviously. I mean, this is in the pond water, right? So it's living matter, right? Bear in mind this afternoon to have also some beads. I have still some 3 micrometer beads. Maybe I'll dilute them a bit so that we have enough for all the experiment. Why am I asking that also we keep some beads is that you will need, either at the end of the afternoon or tomorrow afternoon, you will need to measure some velocity. And potentially some diffusion constant. For this, you need to have a scale bar, and you need to have the time scale. Time scale, it's okay, because time scale, you have it on the microscope, in your cell phone, so you can count the time with your cell phone. Scale bar is a bit tricky, because you're going to have the objective that will magnify, and with your finger, probably you will also zoom. So at some moment, you will not be able to know which scale you have. So I invite you, basically, that we prepare together with Pramod some beads that you will put also in your sample so that you know what is the size of the pixel, right? So that you will have the scale bar, you will have the time scale, and after that, you'll be able to measure velocity in, hopefully, micrometer per second, and diffusion constant in micrometer squared per second. But it's absolutely essential that you measure them with units. You see the point? Is that you need a ruler somehow in your measurement to be valid. You will take your time, take the 4 samples, make many measurements. Don't take only 1 cell. You know, if I were you, I would take 10 cells. It requires patience. I don't know, you know, as you do experiments, in any case, you need to be relaxed. You observe, quiet, right? And you try, you play. It can last 2 hours. Probably the first part will be faster because then you know how to use the foldscope, you know how to couple it with the cell phone, so it will be probably faster. But try to acquire as many examples as possible. If we manage to go fast, then we can start the analysis this afternoon. If not, doesn't matter. I'd rather advise you that you take very careful measurements before rushing to analysis. And it's a general advice that I have for you, whatever you do in PhD or even and after, it's better to have very good data patiently acquired than a wrong analysis crappy, right? So take the data carefully, peacefully, be sure about what you have. That's the best strategy. So now I'm going to summarize what we did this morning. As I said, I wanted to go from, I have 1 more minute before this wrap up. Somebody said this morning, actually, that living matter is directional. I know that when I was asking what the difference between living matter and active matter, whoever that is, doesn't matter. But somebody said, when it is directional, it is living matter, to highlight the difference here. The filament that we saw on motility assay, they were directional. Would you call them living matter? No. In case of motion? But the filaments molecular motors, they are not living, it's a biopolymer, right? So basically, the only observation of a directionality as a signature of living matter is a problem. It's a real problem, right? Because molecular motors are directional, it is not living matter. If you want to call it living matter, then you need to define what is living matter, right? But basically, a directionality is not necessarily signature. Everyone was willing to say worm, filament is not a worm, right? So basically, this distinction here is a very subtle one that requires thinking, and time, right? So, wrap up. What we saw this morning is, again, very different from yesterday. Yesterday is equilibrium physics, KT, Brownian motion, basically given Rho-Reynolds number that every biological system needs to face, cope with. And today was essentially driving out of equilibrium all these species in the biological realm to make the contradistinction with what we saw yesterday. Essentially, active matter, living matter is distinct from inert matter because it uses the chemical energy driven by the ATP hydrolysis or TCG hydrolysis. It drives the system out of equilibrium, gives 10 kT to it, to allow molecular motors to be. a molecular motor is a combination of a polymer and a motor. The interaction between the two objects, which are chiral, the filament is chiral, leads to directional motion, and there is a variety of motors which have all the key functions for cell to be. They can function thanks to this molecular motor and outcompete KT world. Then I showed to you some category of quantification. Duty ratio is one of them. I can mention also that in the way, you can end up with the conclusion that the force they apply is about 10 piconewton. You remember yesterday the estimate of energy over size? If you repeat the energy estimate, replace it from KT to 10 KT, you end up with a 10 piconewton. So indeed, it was measured experimentally. So you have about 10 piconewton. With this, you can shape organism, you can transport, you can read, the same way you can read here the Global Initiative on Academic Network, and you can reproduce this experiment in vitro with purified protein, minimal components. So it's a robust system that is operating throughout the cells. Last word, these elements, basically, whatever you do in every cellular processes you will see, you will find them at play. They are essential. Basically, that makes the physics of living system interesting. With this, I wish you bon appetit. See you later. 2:30 in the other room. Same place. Okay.